All right, so welcome back to another video in our module on the cell structure. And in this video, we're going to continue our discussion with the different organelles in eukaryotic cells. So last video, we talked about the nucleus. This video, we're going to talk about the endomembrane system. And the endomembrane system are the organelles that essentially function like the mail system, the packaging system, the production system in a city. And so, for example, you have your places that will produce different products, and then they will be shipped to be packaged and to be refined, and then they will be shipped out of the cell to be used for other functions throughout the body, or they could remain in the cell and be used for functions inside of the cell itself. And so if we break up the different components or separate the endomembrane system into its different components, it will be made up of the nuclear envelope. So we talked about that structure with the nucleus. The endoplasmic reticulum, which I will refer to as ER, the Golgi apparatus, and then some vesicles, which are lysosomes and vacuoles. And these organelles all are membranous in structure, and they're connected to or linked between one another by vesicles. And they kind of just look like sacs of membranes inside of the cell. And so all these cells interact together by either physically coming into contact with one another or by having vesicles transport products between those different organelles. So here, if we look at a figure of that typical animal cell that we saw in the intro video, we can see all of these endomembrane structures in this figure. So you have the nuclear membrane that comes into contact with the ER. The ER is this purple structure that has the blue dots on it, and then this purple structure that has no blue dots on it. And we'll talk about what that is in more detail, but basically the ER can have ribosomes attached to it, and then another portion of the ER will not have ribosomes attached to it. And so the one with ribosomes is called the rough ER, and then the one without is called the smooth ER. And then you have the Golgi apparatus, which is connected to the endomembrane system through vesicles moving between the ER and the Golgi, and then also the lysosomes and different vacuoles that exist. And so these little round membrane sac structures are the lysosomes and the vacuoles. And you can think about those as like the taxi cabs or the vans or the buses of the cell itself. The plant cell has those same structures, and so it's not too different, except the plant cell has that big central vacuole that we talked about in the intro that helps with kind of cell volume and kind of regulating the size of the cell itself. All right, so the first portion or the first organelle that we're going to talk about in the membrane system is the endoplasmic reticulum, so the ER. The ER is one of the main production facilities in the cell, so you can think of it as any of the manufacturing plants that exist in a city. And the ER develops a couple of different molecules depending on if you're in the rough ER or the smooth ER and the type of cell that it's in. And the ER is physically connected to the nuclear envelope. And so the RNA, for example, could come out of the pore, go directly into a rough ER and attach to a ribosome on it and go to producing proteins within that endoplasmic reticulum structure. And so the rough ER primarily makes proteins and a couple other structures, while the smooth ER makes things like lipids and different hormones. Here we have a zoomed in image of that, where you have that rough ER attached to the nuclear envelope, and then that smooth ER in the bottom right We're also showing where the ribosomes are located on that smooth ER itself. And we call the rough ER that name because it's rough in appearance, it's rough in structure, because it has those ribosomes attached to the outer surface. The ER also is responsible for making more of those membranes. And basically what happens is you'll have a ribosome attached to the rough ER, and it will produce proteins that will increase the size of the ER membrane. Those proteins can be packaged, modified, and transported to other organelles, or it could be transported outside of the cell itself and go to other parts within the body. And so the way that transport happens is there are transport vesicles that will take the products made in the ER and transport them around the cell or even outside of the cell. So again, you can think of those transport vesicles as cabs, cars, taxi cabs, buses, etc. And those transport vesicles will then be dispatched to other locations throughout the cell. Here's a figure where we see those transport vesicles. So basically you have your ribosome making a protein. You see that protein being made. That protein will then be modified, packaged, whatever needs to be done to it before it's transported off. And then that protein will be, so the membrane of the endoplasmic reticulum will actually engulf the protein and kind of form a sphere and pinch off and bud off to make that transport vesicle, which is also why the ER needs to continually grow membrane, because it loses parts of the membrane as it goes into separate transport vesicles. And so then that transport vesicle would move that protein to wherever it needs to go. The smooth ER lacks no surface ribosomes, so it doesn't have any ribosomes on the surface. So since it doesn't have ribosomes, it doesn't make proteins, but instead it makes molecules like your steroids, your lipids, and then the smooth ER is also really important in the liver because it helps to detoxify our cells. So think about it as getting rid of and breaking down those any harmful drugs or any other toxic substances that would be found in our body. Once the vesicles leave the ER, they can then go to the Golgi apparatus. And the Golgi apparatus basically receives those products, refines them, stores them, houses them in a warehouse you could think of, and then distributes those products to the cell. You could think of the Golgi apparatus as like the Amazon of the cell. So basically the ER is the production plant. It gets the information of what to build from the nucleus. And so the ER will build the proteins, it'll build the lipids. And then once those products are made, they'll go to the Golgi where it will be stored or modified or repackaged. And then once it needs to be transported to other places in the cell or out of the cell, it will leave the Golgi to be transported. And so that's like how those two are connected, the Golgi and the ER, and they're not physically connected. They're connected through those transport vesicles that will bring the products from the ER to the Golgi itself. And so up to now, we've talked about a lot of organelles and how they function and work together. And so what I would like for you to do is just kind of take a minute, pause the video, and think about how the following cellular structures could be placed in order in the production line of, say, making and secreting a protein. And so you have structures such as the Golgi, the nucleus, the plasma membrane, the ribosome, and the transport vesicle. So just pause the video and think about how you would order those based off of the production and secretion of a protein. All right, so the order of these would be, so first you would have the nucleus, which was where the information would come from. Then it would go to a ribosome, and then that ribosome would release that protein, and then it would be carried in a transport vesicle, either from the ER or from the ribosome that was free floating to the Golgi apparatus, and then the Golgi apparatus would transport it to the plasma membrane to be released outside of the cell. And that's what we see here in this figure. So we see the Golgi on the receiving side, taking the transport vesicle from the ER. This transport vesicle would fuse with the membrane of the Golgi, releasing the products inside the Golgi. The Golgi will then do whatever it needs to do to that particular molecule. And then as that molecule travels through, it will bud off into a new transport vesicle that comes from the Golgi apparatus. And then that vesicle would travel to the plasma membrane, fuse with the plasma membrane, and release those products outside of the cell if those products needed to be released out of the cell. Okay, and so other vesicles that are found in the cell, an example of one is called a lysosome. And this is a vesicle or a membrane sac with digestive enzymes. And these are only in animal cells. And these lysosomes can break down a large variety of molecules. So they can break down things like proteins, large carbohydrates, fats, nucleic acids, etc. So they basically break down those components into individual parts so they could be potentially recycled into new molecules. Lysosomes are also important for destroying any kind of foreign invaders inside our cells, so like harmful bacteria. And so what they would basically do is take up that bacteria, release the enzymes, and break it down. They can break down organelles to get those individual components again to be recycled to make new organelles. And then they're also important in embryonic development and helping to form certain structures. So for example, like the fingers. And lysosomes are super important because there is also diseases that happen in early development where if in early childhood they don't have the correct amount of lysosomes, this is something called lysosome storage disease, that could be fatal because the body just can't break down those harmful components or the other organelles and molecules that it needs to. Here we have an example of a lysosome breaking down food molecules, and then you have a lysosome breaking down a damaged mitochondria into its individual components. And so you have a vesicle here on the TEM that's breaking down some organelles that need to be recycled. Vacuoles are another organelle that exists, and vacuoles are basically these really large vesicles that have different functions depending on the organism they're found in. So for example, you can have food vacuoles that will come from the plasma membrane, like we showed in the previous slide. Then you also can have vacuoles that can help, say, freshwater protists move water in and out of their cells. And then also things like a central vacuole in a plant helps to maintain the cell volume within inside that plant. So on the left-hand side, we have kind of a vacuole, a contracting vacuole that will move water in and out of a protist, a paramecium, to help that organelle basically move or maintain volume. Central vacuoles in plants, so this blue space here is a very large central vacuole. It fills with water, so it swells, and it puts pressure towards the outside of the cell. And remember, it has a cell wall, so it's rigid and structurally sound. And so it keeps that cell full and expanded. And so when a plant wilts, it's because that central vacuole shrinks and that cell shrinks with it because there's no water inside that central vacuole. And so central vacuoles are also really important because not only is it useful for cell volume, but it also stores organic nutrients and contains pigments that can attract pollinating insects. And so it's really useful for a lot of different things in plants, especially. And so here we have a review of the endomembrane system. So remember, you have going from the nucleus out the nuclear membrane into the rough ER, proteins are made, other molecules are made, transported to the Golgi where it's refined, packaged, and then the Golgi transports it either inside the cell to other places or outside of the cell where it will be used in different locations in the body. All right, so that's it for our discussion of the endomembrane system. We will carry on with our discussion of cells and different organelle structures in the next video.